Hello all, how you doing? I'm your instructor for Bio 151, which is Biology 2. Hopefully you figured out how to pronounce my name. And if you said Dr. Buoni, you are correct. This is a short video to kind of give you an outline of the course and also more particularly what you'll be doing the first week. I have had this absence planned for a long time, so I will not be there just the first week of class, but it doesn't mean you don't need to keep all the work going. So this video will not only show you what to do this week, but will explain the course and how it works. I will be back week 2 all the way through the rest of the course. So when you get into the course, it'll look something like this. There will be an announcement, but just a couple things about the way the course works. This course is hybrid. You signed up for a hybrid class, and what that means is that most of the lecture work, the studying, happens on your own time at home. A very large percentage of the time that you spend in class is going to be dedicated to lab activities. That's what a hybrid class is. And so therefore a lot of the work that you have, a lot of the assignments and actually your exams are taken online through this thing called Pearson. So you will need a Pearson access code as indicated in the bookstore to do not only the work for a lot of the assignments but also the exams. So be sure to have that, and I'll show you how to access that later. So first things first, what's this look like? So we hit content. Now yours will look a little different than me because you are a student, but everything that you need to see you will be able to see at this point. In other words, you might not be able to see all the way down to the end. That's fine. The first thing you're going to do is start here, and obviously there's a video right here that you're watching, so you already know to do that. And this is the video that you'll be watching. Here is information about me. This is my email. Obviously the best way to contact me is through my email, at least 2 days, not including weekends. If you want to know what you need for the course, you can go here, student support, etc. So let's get down to the policies. This document talks about how long I get for communication, the things you're going to need. Here is the book. Again, if you want a paper copy of the book, that's fine, but the more important version is what you need with the Pearson Mastering access code, which also gives you access to an online version of your book as well. And you can read all these policies about everything in the class. This is very important, this part. You have to get at least a 70% or higher in lecture and a 70% or higher in lab separately to pass the class. So if you happen to have a 90 in the lecture and a 68 in lab, even though if you were to take the average of those you pass, you actually don't pass. You have to have a 70 separately in both the lecture and lab portion. Here is the grading itself, and you can see here wow, I have these exams, they're worth 70% out of the 75 of the lecture, and these MyLab mastering assignments are only worth 5. Well, why would I do that? Well, the answer is this 5% completely prepares you for this 70%. So that's why you want to make sure that you're doing all those work. The lab grade is based on a series of, first of all, your lab notebook. We'll talk about that when I get back in week 2. And then lab quizzes and lab reports, and so that is 25% of your grade, and this is how everything is broken down. So you can go ahead and read all of this all the way down, and that's great. So if you want to read the syllabus, you can go click here and it'll send you right to the syllabus of the course. You can click that. I don't need to do that here. You can do it on your own. What I do want to spend time on is the course schedule. So let's take a look at that. So if we open up the link of the course schedule, all right, so what do we see here? We basically see that this is kind of like your schedule for the whole 15 weeks, so you know exactly what you have to do. Now, understand here, I will not be there this week. You do not show up this week. You don't need to show up this week. I am actually doing the introduction of the course, the expectations and all of that stuff right now. We're going to talk about your this is all covered in your week 1 stuff. You don't need me for it, and you do actually have an assignment you're going to do that is due on Sunday the 19th that you're going to do in Pearson Mastering Biology. So that is what we're doing this week. So as we move down the schedule here, The other part that might seem weird is like, well, wait a minute, like, so I don't get to meet you, Dr. B, first week, and it looks like we also don't have class the second week. No, we don't. The reason why is that there's some sections of this class that happen on a Monday, and that is Martin Luther King holiday, so there is no school for them, so there is no class for us. So I won't meet you actually until the 27th, which is fine. So you have all of this work to do by yourself, which again, making it sound like a huge amount. It's not a terribly huge amount. Which is going to be this on week 2, using an NCBI assignment. And again, I will be back around in town. The reason I will not see you this first week is I've had a trip planned for a very long time, and I happen to be gone the first week of the classes. So I will be back and engaging. So if you have any questions during this week, I will be available via email, but I just won't be there in person. So this allows you to kind of make sure and prepare what you need to prepare for each of the weeks as we go down, and you can kind of see what's due when. So obviously the biggest part is going to be the assignments. Here they are. And then you have your exam. Your exam, by the way, is online, also through D2L. And so that is a great way for you to kind of organize what you need to organize for the course. All right. So we have that. We have the MyLab Mastering Resources. So there's a couple things. I'm going to kind of go through a couple other things that would matter first. Now, Digital Lab Notebook, you don't need to worry about this until I get back into town. So we won't even deal with this until I'm back. Let's just deal with week 1 and week 2, which are the 2 weeks that I won't be here. Or actually, I will not be here just the first week, but we don't have class the second. So how is every week organized? Well, basically, you have an overview. And here is the overview of what this week 1 is about and what you have to do. And you have to register for MyLab and Mastering, and you literally just click the button. Of course, it's going to say no page found. That's because I'm not a student in the class, but it will work for you. And then every week you also have what's called Read, Study, and Practice. So this is in essence the major content of the lecture portion of the course, obviously besides the textbook. So, here's one, here's a little bit more, how do scientists organize biodiversity? And then from that point you have an assignment. And notice the assignment says Organizing Life on Earth. That also matches up with the actual schedule itself, because look what you have week 1, Organizing Life on Earth. Then we just repeat. Week 2, there's your overview about what week 2 is about, which I love that, by the way. And what assignments do you need to complete? And here's again the Read, Study, Practice. So this will give you the major aspects of the content. And here's a second one, and that you would be basically taking notes on this type of stuff. Also looking in the textbook as well for backup information to it as well. And then these are the assignments. This one here is using NCBI, which is an international database. And you can see here that there's a video, by the way, that's going to show you how to do all this. And I will, of course, be back in town for this. So that's kind of how the course is organized. But when I see you in class from weeks 3 all the way down to 15, most of the time we are doing lab activities. And if I go back to the schedule for a second, the lab is based on 2 really big projects. We at Delaware Tech pride ourselves on doing authentic undergraduate research. So this is not like high school where you had 1 week you had a different lab, for the whole year. We don't do that. So we have 2 big lab experiments. So one of which is going to be a study on campus of the tick density. Ticks that cause Lyme disease, and by the way, a myriad other diseases as well, or they carry them. So the first couple of weeks we're going to show you how to go out in the field. Bio 151 is a very ecology based class, so we're going to go out in the field. We're going to talk about how exactly we're going to collect ticks and be able to discuss the density of them, record all the data, and then we're going to actually isolate DNA from them. So that's going to be a session we're going to do in the early spring. And then if you notice down here, we also repeat that down in April. We want to see if there's a statistically significant difference between the density and species of ticks that we collect in the winter versus early spring versus later on in April. Ticks are actually found because of lots of things, climate change being one, or at least local weather patterns, both of them. We do find ticks pretty much all year round, so we want to see if it varies what kind or species and the amounts that we find. So that's going to be the one lab that's going to start in the beginning, and then we're going to pick it up again at the end. The other lab we're doing is here, and the bulk of the middle of the course is going to be talking about a lab that is, excuse me, being designed and tested in your class, which is looking for the snakehead in freshwaters of Sussex County. So this is actually a picture of a snakehead fish, and you can see why it's called the snakehead fish, because its head looks a lot like a snake. A lot of people consider it pretty ugly looking. But it's an invasive species of fish, and what makes it terrible is that it's an apex predator and that it can outcompete other local fishes and also aquatic life, which is bad. And the other thing it has is this amazing ability in times of things like drought that it can actually come out of the water and quote unquote walk over to other bodies of water in times of drought or stress or whatnot. And it's definitely an invasive species. So with that, are we going fishing? The answer is no. We are going to attempt to filter water through filters from these water sources, and every time we have this filter that catches any sort of organic material, we're going to then isolate the DNA from these filters after we run water through it for like 45 minutes, and then we're going to look for DNA from the snakehead. So for example, like if there is a snakehead, obviously some of its scales might fall off, or some of them might have died, and little teeny pieces of DNA from the fish could be in the water. So it's a way for us to identify whether a given body of water has or had a snakehead in it based on looking for it using molecular biology, which is really cool. So that's the middle lab we're going to do. So this again is just a kind of short overarching introduction, enough to get you started till we see each other again. So again, if you have any questions, please let me know, and I'm here to help. And welcome to the class.